Hello, Smart Medicoz, and welcome to the Deep Dive. Ever stop to think about the incredible silent war that's going on inside your body, like right now?
It's pretty amazing stuff.
Yeah. Today we're diving into one of your immune system's most basic yet powerful processes, phagocytosis. Basically, how your cells literally eat invaders and, clear out cellular junk. We're looking at the hidden heroes, tackling what the textbooks call offending agents.
Right. And what's really crucial, I think, is that this isn't just about fighting off bacteria or viruses you catch.
Okay.
It's also absolutely vital for just, cleaning up, clearing out old or damaged cells. It's fundamental for just maintaining your overall health.
So like cellular housekeeping too.
Exactly. It's a very precise, very powerful operation happening constantly.
Okay, let's talk about this cleanup crew then. The main players, neutrophils and macrophages.
Those are the big ones, yes. Neutrophils and macrophages, think of them as the frontline responders.
Right, and their job is basically eat and destroy. Microbes, dead tissue, that sort of thing.
That's the core function, ingest and destroy. But importantly, it's not random. They don't just go around eating anything.
Right.
No, it needs specific activation. Think of it like flipping a switch. You need certain signals inside the cell, increases in calcium, activating enzymes like protein kinase C, phospholipase A.
Okay, so signals get them ready for action.
Precisely. And once they are activated, it leads pretty directly to two main things. Actually engulfing the target, the phagocytosis part, and then
Destroying it.
destroying what they've just eaten, yeah. Intracellular killing.
So how does that eating part actually happen? You described it as almost like a ballet.
Well, maybe a very aggressive ballet. It's a tightly coordinated sequence, definitely. 3 main steps.
Okay.
Recognition and attachment. The phagocyte has to identify its target first. It uses specific receptors on its surface to bind to the particle, whether it's a microbe or debris.
So it knows what to grab onto.
Exactly. Then comes step 2, engulfment. This is quite dramatic, actually. The cell membrane extends out and flows around the microbe, zipping up, essentially.
Zipping up.
Yeah, forming a little internal pouch or bubble containing the target. That bubble is called a phagosome.
Phagosome. Got it. The final step.
Killing and degradation.
Yeah.
That phagosome, with the target inside, then fuses with another internal compartment called a lysosome.
Lysosomes. Those are like the cell's recycling centers, full of enzymes.
Full of very powerful digestive enzymes, yes. When they fuse, you get a phagolysosome. And inside that, the target gets broken down, destroyed.
Wow. Okay, back to that recognition step. How do they know what to eat? You mentioned receptors.
Absolutely key. They have a whole toolkit. There are mannose receptors, for instance, which bind to sugars often found on microbes. Scavenger receptors bind to things like oxidized LDL, bad cholesterol, but also microbes. And integrins, like one called MAC1, also help them grab onto targets.
So different receptors for different kinds of targets.
Right. But maybe the most important ones are the opsonin receptor.
Opsoning.
Yeah, opsonization is a crucial process. It's where your immune system essentially tags an invader for destruction.
Tags it how?
With molecules like IgG antibodies or complement proteins like C3b, or even things like mannose-binding lectin, these tags, or opsonins, stick to the microbe.
I see.
And the phagocytes have receptors specifically for these tags. Binding to an opsonized particle is much more efficient. It massively enhances phagocytosis.
So it's like putting a bright eat me sign on the bad guys.
That's a perfect analogy, yes. Makes the whole process much faster and more effective.
Okay, makes sense.
Yeah.
So once they've engulfed it, tagged or not, what's the actual killing mechanism inside? You mentioned enzymes, but it sounds like there's more.
Oh, yes. They have a pretty potent chemical arsenal. 2 main systems really stand out. First, reactive oxygen species, or ROS.
ROS sounds dangerous.
They are highly reactive molecules. The phagocyte generates what's called a respiratory burst using an enzyme, NADPH oxidase. This churns out things like superoxide, hydrogen peroxide.
Hydrogen peroxide, like the stuff you put on cuts.
Sort of, but generated right where it's needed. And there's even a specific system involving an enzyme called myeloperoxidase, or MPO, that uses hydrogen peroxide and chloride to make hypochlorite.
Hypochlorite. Is it basically bleach?
Chemically, yes. Bleach, generated inside the cell to kill microbes.
Wow. Okay, what's the second system?
Nitric oxide, or NO. Especially in macrophages, an enzyme called inducible nitric oxide synthase, or INO, produces NO.
Nitric oxide, I've heard of that in other contexts.
It has many roles, but here it can combine with that superoxide we mentioned earlier to form an even nastier molecule called peroxynitrite. Very toxic to cells.
So a double whammy. ROS, and NO derivatives. Where are all these chemicals and the enzymes stored before use?
They're often packed into granules within the cell. Neutrophils, for example, have different types. Specific granules with things like lysozyme and lactoferrin, and azurophil granules packed with MPO, defensins, elastase, acid hydrolases.
A whole cocktail of destructive stuff.
Exactly. Macrophages also have granules with acid hydrolases and other enzymes. Now, these are great for killing things inside the phagolysosome, but if these powerful enzymes leak out or are released outside the cell, they can damage healthy surrounding tissue. That's a potential downside.
Is there anything to stop that?
Yes, thankfully. Our bodies have antiproteases, like alpha-1 antitrypsin, circulating specifically to neutralize enzymes like elastase if they escape. It's a control mechanism.
A built-in safety net. That's clever. But I read neutrophils have another trick up their sleeve, something that doesn't even involve eating.
Ah, yes. You're thinking of neutrophil extracellular traps, or NETs.
NETs? What are those?
It's quite extraordinary. Neutrophils can actually eject their own DNA, their chromatin, which unfolds to form a sticky web outside the cell.
They throw out their DNA.
Mixed with proteins from their granules, yes. These nets physically trap microbes, and the associated enzymes can kill them right there in the web extracellularly. It often involves that ROS generation we talked about to decondense the chromatin.
That sounds like a pretty drastic measure.
It is. It's often seen as a sort of last resort. And there's a big trade-off. The neutrophil dies in the process of forming a net. It's a form of cellular suicide to trap pathogens.
Wow. Any downsides to that besides the neutrophil dying?
Well, sometimes releasing all that nuclear material and cellular content can expose bits of ourselves, autoantigens, that the immune system might mistakenly react to later. There's evidence linking NETs to the development or exacerbation of autoimmune diseases like lupus.
So it highlights that complex balance again, helping fight infection, but potentially causing other issues.
Exactly. Which brings us to the unfortunate reality of leukocyte-mediated tissue injury.
Right, the double-edged sword you mentioned. When does this collateral damage happen?
It can happen in several situations during really persistent infections like tuberculosis, where the immune response is prolonged and intense. It's a major factor in autoimmune diseases, where leukocytes attack the body's own tissues, and also during severe allergic reactions or hypersensitivity responses.
And the mechanism is basically leakage.
Pretty much. Those potent ROS, NO, and protease we discussed getting released into the extracellular space, either accidentally or sometimes deliberately. There's also a phenomenon called frustrated phagocytosis.
Frustrated?
Yeah, imagine a phagocyte trying to engulf something really big, like an artificial implant surface or large crystals. It can't swallow it. So it gets frustrated and just releases its destructive enzymes outwards onto the target and unfortunately onto nearby healthy tissue too.
Ouch. So beyond just eating and killing, what else are these leukocytes doing during inflammation?
Oh, they're multitaskers. They're crucial sources of cytokines signaling molecules like interleukin-1, IL-1, tumor necrosis factor, TNF, IL-6. These really amplify the inflammatory response, calling in more troops.
So they coordinate the battle.
Absolutely. They also release growth factors that are important later for tissue repair and enzymes that help remodel the damaged area. And don't forget, other immune cells interact with them, like Th17 cells, which produce IL-17 specifically to recruit more neutrophils. If you lack Th17 cells, you become very susceptible to certain fungal and bacterial infections.
It's all interconnected. So how does this intense process, acute inflammation, actually end? It can't just go on forever, right?
No, thankfully. Several things happen. The initial stimulus, the infection or injury, gets removed. The inflammatory mediators, those cytokines and other signals, have short half-lives, so they naturally degrade. And neutrophils themselves are very short-lived cells. They die off after a day or so in tissues.
So it just sort of peters out.
Partly, but there are also active termination mechanisms. It's not just passive decay. There's a really interesting switch from pro-inflammatory lipid mediators like leukotrienes to anti-inflammatory ones called lipoxins.
A chemical switch to calm things down.
Exactly. Plus, anti-inflammatory cytokines like TGF-beta and IL-10 get released, actively suppressing the inflammation. And even neural pathways, believe it or not, can get involved, like signals through the vagus nerve inhibiting TNF production. It's a highly regulated process, turning both on and off.
That's incredible complexity. Okay, final thought, Ben. Considering just how powerful and intricate these self-defense mechanisms are, this whole phagocytosis process, what does it really mean for your health when these finely tuned cellular processes, this eating and cleaning, go wrong? And looking forward, what new doors might this deep understanding open up for medicine?